today for our research seminar given by Chrysanne DiMarco. She's a professor in the school, I should say, the David R. Cheriton School of Computer Science at the University of Waterloo. Before we start, I just have one announcement. We also hold another seminar series called the Waterloo Smarter Health Seminar Series. This year it's devoted to the university's anniversary theme of Why Not? And our guest speaker, who will be speaking on May 23rd at 3 o'clock, right next door in room 1302, is Dr. Jeffrey Fong. He'll be speaking on Why Not Evidence-Based Health Policies for Nations, the Case for Global Tobacco Control. Jeff is a world expert in this area. He's a dynamic speaker, so if you have any opportunity of coming out to hear this seminar, please do so. For those of you that might see this presentation on the web, we will also be broadcasting this on the internet and through video conferencing to the OTN telehealth network sites. So without any further ado, Maher, will you please introduce our speaker?
Good afternoon, everyone. It's my pleasure to introduce our guest speaker today, Chrysanne DiMarco, Professor of Computer Science and member of the Artificial Intelligence Group in the David Cheriton School of Computer Science at the University of Waterloo since 1990. Her research interests include computational models and, of natural language pragmatics, computational linguistics, health informatics, and biomedical information extraction. Professor DiMarco is the project leader of the HealthDoc project since 1994, which has been developing web-based natural language generation systems for producing health information tailored to the medical conditions and personal characteristics of an individual. Also, she is the president of Inkbot Software Inc., the spin-off company from the HealthDoc project. Today, Dr. DiMarco will talk about where computer science, linguistics, and biology meet, using lexical chaining to analyze biomedical text. Welcome, Chris.
Thank you, and thanks to all of you for being here. The work I'm going to describe has actually allowed me to fulfill a dream that I've had since first year university, where I couldn't decide whether to major in English and French or mathematics and biology. And as I went on through university, my path diverged and I ended up in computer science. But in recent years, I've been able to come back and combine all my interests, because in the meantime, this new field of biolinguistics has emerged, and I'm going to describe some of the work our group has been doing in this area. A particular problem we've been working on is trying to get information out of the now a huge volume of online literature in scholarly scientific articles. One particular problem that people are working on is trying to extract information automatically about biomolecular interactions. And protein-protein interactions is a topic of particular interest. Why biologists want to find out about protein interactions is because from knowing about these interactions, it helps them discover more about the biological functions of the proteins involved. So there are protein interaction databases, but when we went and met with biochemists working in the protein interaction field, the first thing they said to us was these databases were often not very useful to them because a lot of the information in the databases is spurious. So information has been extracted from online articles that purports to be a valid interaction, but it really isn't, because sometimes interactions between proteins will occur under laboratory conditions, but they don't actually occur in the living cell. And so to biologists, this wrong information can be worse than none at all. For a biologist to try and read all the papers available, even on a single interaction, there's just hundreds of them, so there's no way they can try and keep up with them. So this is a prime area for the use of computers in enabling people in the real world to do their job better. If we can create automated methods for biomedical information extraction, we can extract information that human beings can't. And our particular niche in this area is to try and validate the interactions once they are detected. So what we are going on is knowledge from biology, biochemistry, that proteins that are involved in interactions will tend to have similar biological functions. So being linguists, computational linguists, we thought, well, if that's the case, if proteins that interact have the same functions, you'd expect that when these interactions are described in an article, that you'd find biologically similar terms in the context surrounding them. And if we can detect that, then that should be evidence that that interaction is in fact valid. So in order to find those biologically similar terms, we are relying on a method from computational discourse analysis called lexical chaining, which, just as it sounds, builds strings or chains of semantically related terms in a given context. Now, the work that's going on in biomedical information extraction, this area has really just taken off in the last few years, and it turns up as a theme at major computational linguistics conferences. There are now evaluation tasks going on in this field, but the natural language methods are still pretty simplistic. They rely on partial syntactic analysis, little to no semantic understanding, and really what people are trying to do is just even finding a token, really finding out what is a word, what is an acronym, what is an abbreviation, isn't still an extremely hard problem. Part of speech tagging, trying to find out what a token or a word is it a noun, is it a verb? And shallow parsing means just trying to find the overall structure of a sentence. Where's a noun, a noun phrase, where's a verb phrase? So because these articles are lengthy, because there's so many of them, you can't do detailed semantic understanding. So at the most, what people are doing is using what's called a template-based approach, and if they can take a sentence and dissect it and say something like, protein A interacts with or binds to or associates protein B, then that's what they use to extract information that these two proteins are in fact related. But this template-based analysis, and it's ironic because this goes back really to the dawn of computational linguistics when these very simple methods were tried. And we've come back to them now because we're faced with huge volumes of text corpora. Of course, as you can see, it misses a great deal of semantic information, and we'd like to do better. So we wanted to use computational discourse analysis, but in a finessed way. Try to get as much semantic meaning, but in a computationally tractable way. So lexical chaining was introduced by Morris and Hurst in 1991. It was actually Jane Morris's master's thesis in 1988. And it comes from the linguistic concept of textual cohesion, that when people write a text, they don't just throw words at random, but a text is composed to stick together, to form a cohesive, unified whole. And there are different ways of making a text stick together. By reference, we mean things like pronouns. If you have nouns in a text and pronouns that refer back to them, there's a sticking together of those two constituents. A substitution means words like one. Like, I have a bottle of water, and Shirley has one too. The one and the bottle of water relate together. Ellipsis, funnily enough, when we leave things out in the text, cognitively we know that there's meant to be something there, and it refers to something back in the text, and so on. So these are syntactic forms of cohesion. What we're looking at is actually lexical cohesion or lexical semantic cohesion. What are relationships between words or concepts that we can use to build up an understanding of the meaning in a text? And there are some very classic semantic relationships, like one word is a part of another word, like car and motor. One word is more generic, like jaguar and automobile. So a lexical chain, formally, is a sequence of words that are related by these classic relations within a span of text. And a document contained many, many lexical chains. And if we could find them all, they'd give us an indicator of the topic structure of the text. So that's what we're looking for. So this is an example of a little lexical chain. John has a Jaguar. He loves the car. John works in the garage taking care of his Jaguar. So the lexical chain are the words marked in blue: Jaguar, car, garage, Jaguar again. And the relationship between Jaguar and car is what's called hyponymy. Jaguar is a specialization of the more generic term car. And words like car and garage, or garage and jaguar, are collocations, meaning they tend to occur frequently in the same context. Also, the occurrence in the lexical chain of jaguar and jaguar is a valid relationship. It's a repetition. So if we can find these kinds of relationships in biomedical articles, the ultimate goal is they'll help us understand the meaning of the text without going to deep semantic understanding. So people have developed lexical chaining algorithms since the late 1980s, and this is the algorithm for a very simple one. What it means is, though, you have to build all the possible connections between words in a text. So people have focused on building nouns because those are what we tend to understand best, and we have online lexicons for nouns. But these simple lexical chaining algorithms were not very tractable. I mean, in computer science terms, they're not very efficient. And when we're dealing with corpora, which are huge, trying to build all possible lexical chains and then figure out which ones are really good it's not very useful to us. So Greg Silber and Kathy McCoy came along with a linear time algorithm, which to non-computer science, it just means it's much, much better. It's something that we can actually hope to use in practical applications. And what they did is they only built lexical chains which looked like they might be good, and then they used a very simple scoring mechanism to winnow out the ones which they didn't think were practical. And there have been some recent improvements. One of my students, Matthew Enns, he tried out a very interesting application. For a long time, it looked like lexical chainers couldn't be improved upon. But what Matt thought is that if you could disambiguate word senses, which means that words can have more than one sense. Like the word bank can mean the bank of a river, or it can mean the place where you go and take money out. If we can figure out the particular sense of a word in the context it's being used before we do lexical chaining, then the lexical chaining algorithm should build much better lexical chains. So if we're working in a biomedical context, if we can throw away all the meanings of a word which are non-biomedical, our lexical chainer should build better lexical chains. And in fact, that's what Matt was able to show. But he also showed that in some cases, when you did this perfect word sense disambiguation, you missed out on some possible chains. So, but in any case, it's Matt's improved lexical chainer that we used in our experiment. Just in passing, I had mentioned to you what people are doing now in information extraction, and we've adopted this formula for the work we're doing in biomedical information extraction. And really the thing I want you to get out of it is that right now people are doing what's called very shallow methods. Because of the difficulty of extracting information out of complex text and volumes of large text, you can't do deep semantic analysis. So at the very best, you try to find something like a template. There's a noun phrase, there's a verb, there's a preposition, there's another noun phrase. And if something doesn't fit that template, it's very difficult to get at its meaning. So we want to do much better than that. So our ultimate goal is to take a lexical chainer, combine it with an information extraction algorithm, and use that to get at the meanings of these protein interactions in biomedical articles. But what we almost need to do is define a stylistic grammar for biomedical writing. We need to understand how do people write in this context. We know that from genre to genre, people compose text in a certain way. And computational linguists, funnily enough, have tended to work on what's very self-referential. They study their own writing. And computational linguists are actually very bad writers. And they don't write things in a very structured manner. So when you look at biology or chemistry or physics articles, they're written in a much more formal and scientific style. So we've learned a lot from looking at material in the rhetoric of science, and we have a consultant in that field working with us. To give you an idea of the complexity of the kind of language we're dealing with, this is a sample protein interaction lexical chain taken from an article. And to start with, I did a manual analysis of about 20 articles on protein interactions. Now, some of these articles were 20 pages long, and there were dozens and dozens and dozens of protein interactions mentioned, and building these lexical chains was no mean feat. And some of the words that appear in this genre are the meaning is quite different from what you would expect. For example, the words that are in blue, mutations, defects, that's a lexical chain, because a mutation in terms of a protein interaction is actually considered a defect. But in another context, a mutation might actually be a good thing. It might mean an evolution towards being a better and kind of entity. So you have to understand the context to see what to put together. The words in green, cell growth, cell cycle control, telomere length, regulation, those are all related, but it requires becoming a domain expert, almost becoming a biochemist, so that you can understand that these terms have a contextual similarity. And so even doing that small sample of 20 articles taught us that what we were missing, and that leads into the big aha, is that we didn't have a way of understanding the words that were in the articles. We didn't know when things were related, how they were related. That was we had a solution, but we were missing this big resource that how do we understand the words in these articles? So I'll just mention in passing that what we hope to do eventually is build the lexical chains, figure out how strong they were and how good they were, and that the quality of the lexical chains is going to give us some evidence about the goodness of the protein interaction. And there hasn't been very much done up till now on scoring lexical chains. The linear time lexical chaining algorithm I'd mentioned, they had a very, very simplistic scoring mechanism. Work by Graham Hurst and David Saint-Onge improved upon it a little bit, but there's, again, a lot more to be done on exploring the nature of measuring the quality of a lexical chain, and it may vary from genre to genre. So I'm just putting that up to say people have considered it, but not very much. So this is where we were at, and I guess about 2 years ago now, we wrote a paper for the International Systems on Molecular Biology conference, and we got back enthusiastic rejections. In fact, I think they were the best rejections I'd ever received. I mean, they were much more positive than many acceptances I'd received. That this is novel work, it's exciting, it's creative. But what was missing was, ta-da, an ontology. That there was no dictionary of the terms that we needed. How were we going to build these lexical chains when we didn't know the relationships between the words in protein interaction articles? Because there was no lexicon for the protein interaction domain. What lexical chainers have used is a by now standard linguistic ontology called WordNet. It's a online thesaurus of standard English. I forget how many tens of thousands of words in WordNet, but it was painstakingly created by hand by linguists, and it relates words by common semantic relations like part of and specialization, synonymy, antonymy. Words are grouped together they're called synonym sets, synsets, and a synset represents a group of words that have a close near synonymic relation. So that's what lexical chainers need to work on, but there are no word nets for the protein interaction domain. In fact, there are none for biology. So we had to take a giant step back and for the last 2 years create this ontology. In fact, that's what the reviewers said from the ISMB. Well, this is a great idea, folks, but there's no ontology, So we have spent a lot of time, we've had this great idea to start with. We've spent a lot of time creating these necessary resources to get back to the key idea we had in the beginning. So that's what we've been working on for the past year, is creating an ontology, a WordNet-like ontology for the protein interaction domain. And we started out with the Gene Ontology, which, as you may know, is a very commonly used biological ontology. And it had some nice properties, like WordNet. It's a hierarchy composed of, again, basic semantic relations like part of, and in the Gene Ontology's case, associates with. And it describes gene and gene products in any organism, so it's got some things we need. The ontology is divided into three components: biological processes, molecular functions, and cellular components. But, and I should have put this in blinking red, it has nothing about protein interactions. So although here's an ontology that people in the biomedical community recognized, and the unified medical language system is there as well, but they don't contain terms specific to protein interactions. So what to do? Well, this became the master's thesis of Xiaofen He, one of my students who graduated in January, to take the Gene Ontology and create a PPIWordNet. So what you have here is some of the terminology in the Gene Ontology. And we had two problems. First, what were the terms that we needed to identify the protein interaction domain, and where would we fit them in to PPIWordNet? How would we do that? Now, my colleagues in artificial intelligence have the grand vision of being able to build ontologies automatically. After the next few minutes, I'll leave it to you to decide when they might be able to do that and how feasible that goal is. So Xiaofen's work, the major piece of her work was coming up with a protocol for constructing PPIWordNet, and several people were involved in the process. We were very lucky to have experts in several domains. Shadi Hassan, who is a PhD student in engineering and works in information retrieval, he did the information retrieval part of the work in extracting out what are called the discriminating terms, the concepts specific to the protein interaction domain. And then we were very lucky to find two PhD students in biochemistry at the University of Toronto who were willing to learn what concepts like ISA and part of and construct ontologies and all that sort of thing, Gabe Musso and Zhou Yu, and they hand-built the ontology and fitted it into the Gene Ontology. And then Xiaofen integrated their concepts with the Gene Ontology and tested it out. What was the proof of the pudding? How well would this new PPIWordNet work in lexical chaining on biomedical text and protein interaction text? Just to determine what were the key concepts and relationships in the protein interaction domain, there aren't a lot of materials available, and how do you go about this? And Xiaofen chose a middle-out strategy, which was find the core concepts and then specify and generalize as necessary. So we tried to be as systematic as we could, but we found out there were times when we just had to rely on the domain experts' knowledge. They had a feeling for there were terms that they expected should be there because of the reading they had done, and they would put them in as necessary. We wanted it to rely on a corpus-based methodology, as is done commonly now in computational linguistics. So we wanted to be able to extract out of existing large corpora of protein interaction texts the terms that were actually used. That became a real issue. In fact, it held us back for about a year because this is an instance of how raw this field is. There were no corpora of full-text protein interaction articles. In fact, William Hirsch, when he was here as a guest of Dominic, was very generous in allowing us access to a corpus of 10,000 general biomedical articles from which Xiaofen curated any article that mentioned protein interactions. But, and then she scored them for the ones that seemed most likely to be relevant to us. But we were picking up articles which might say, once in the article, protein A interacts with protein B, meaning that these papers were not necessarily about protein interactions, so they were really not very good for our purposes. But these full text articles were just not available, and maybe this is something we could speak to after my talk, about for copyright reasons and about the non-accessibility of these articles, why it has slowed down research in this field. But just this last summer, I went to the Computational Linguistics main conference and picked up a leaflet. It was one of those serendipitous moments, and saw that an organization called BioCreative was sponsoring an evaluation task on detecting automatically protein interactions in full-text articles, and as part of the evaluation task was going to be making available to everybody these gold standard corpora of full-text articles. So if I hadn't gone to the conference and if I hadn't been wandering around picking up pieces of paper, I would not have found this out. So that's how we got access to these texts. And so finally we had the material to work on. So that was our task corpus. I could tell you a lengthy story, and Shadi, if he was here, could tell you how laborious it was, because this was just to show you what researchers go through. This was sort of the grungy bit of the work. We got these full text articles, but they were not in a form we can use. And I know this is may not sound very exciting, but it held us back for months, just stripping out, and getting the markup and getting into a form that we could analyze. I mean, see, these are sort of the basic things that that they get in the way of doing the really interesting work. But it must have taken us 3 to 4 months of working on this corpus before we could actually start to use it. And what we wanted to do was to compare the biocreative corpus with Wikipedia, which we took as a corpus of standard English, and compare the two and see the terms that didn't appear so frequently in Wikipedia, but did appear in the biocreative corpus. Those should be symptomatic of protein interaction terms. And we ended up with about 13,000 terms, most of which were useless to us, because we got things like the author names and abbreviations, and we really had to rely on our domain experts to hand select the terms. We came up with about 200 terms. And then the interesting part of this work began. How do you create a WordNet-like ontology? How do you organize these terms into synonym sets? And this is where you really have to rely on the domain expert. This is where I think that automated ontology learning, I'm sorry to say, I don't think machines will ever be able to do this in the way that human experts do. And so there were months of work in just taking these terms and organizing them into synonyms and building the relationships between them. And along the way, the domain experts quite happily became knowledge engineers. So there was a real interplay between the computer scientists, the computational linguists, and the biochemists and the information retrieval people. I think it was very necessary that we had a group of people who were all experts in their field and who all contributed to this task. And it was as systematic as we could make it, but there were times when we just had to rely on the experts to say, you need this concept here. This is the way it's related. And then they would show us their ontology, and I'd say, well, this doesn't look quite right. And there was a very good interaction among all of us. So I don't think you can see this too well and read the terms, but what I want you to look at really is the structure. This, if you look at it, it's a bit of spaghetti code. This was the first ontology that the domain experts built, and my impression is that a lexical chainer is going to have a lot of difficulty doing some form of automated inferencing. One, there's a lot of connections. It's very deep. It's kind of a lot of branches. It just didn't look very clean and neat. We wanted something more machine-like. So they refined the ontology, and it was really just a matter of cleaning it up and making it, I would say, more formal. Trying to classify the relationships between concepts in terms of relations that people in knowledge engineering understand most well, like the is-a relationship, the part of relationship. And so then what we came back to was this. And even if you can't read the terms, you can see that it just looks cleaner. It looks more systematic. And we only have about 3 levels down, and that turns out to be enough, because a lexical chainer has to recognize that this is a specialization of the top concept. This is a specialization of the one at the second level, and so forth. So you actually don't want more than 3 or 4 levels. And then from what we had done to that point, we integrated, or Xiaofan integrated, with the help of the domain experts, our concepts with the gene ontology, and again, trying to do it in a systematic way and hooking things into the gene ontology where they seem to fit best. Okay. And now at that point, we had our PPIWordNet. Now we wanted to see how well does it work. Does it allow us to do some practical natural language processing task and do it better. And we're still on the road to being able to eventually see whether protein interactions can be validated using lexical chaining. And our hypotheses were that we could do deeper discourse analysis using lexical chaining than these current biomedical information extraction methods, which are typically very shallow. But we also had to prove, and that was something that the ISMB reviewers had said, I mean, do lexical chains actually occur in protein interaction articles? I mean, that's something we've been assuming. Maybe they don't even exist. So we had to show that. So we took 100 full-text articles from the task corpus, and then Xiaofen went step by step. First, she used only the Gene Ontology, and then she started adding in components of our protein interaction WordNet and seeing how it affected the lexical chains at each step. We also came up with some metrics. We wanted to simply see how many lexical chains there were in an article, and then the quality. We figured that the longer the chain, the more important the topic, and the more dense the chain, meaning the more terms in it, you know, the more important the topic. And along the way, we said to ourselves, well, there's some way we need to decide how rich a chain is. That if you have if you have all the same word repeated, well, that might be a reasonably good chain, but it might not give a lot of evidence about the quality of the protein interaction. So the more terms in a chain, those are called lemmas. You know, the number of mixed terms in a chain would actually be greater evidence for the biological validity of an interaction. Okay. So these are the results of the experiment. This was the culmination of Xiaofen's master's thesis. So as you can see, the number of terms in the articles, as we added from the Gene Ontology, went up a bit. But the number to look at here is the number of chains. With the Gene Ontology, there were about 4,500 chains. When we added in just a few more, it went up to 5,000, a few more to 6,600, and then the final number of concepts, almost 5,900. Now keep in mind that the Gene Ontology itself is about 60,000 concepts. We were adding in less than 100. But they were extremely targeted, and they were put in places in the Gene Ontology where it really mattered. So even less than 100 terms made a big difference. Some of the other statistics to look at, the number of concepts in a lexical chain didn't vary all that much. So this average lemmas means that most of the terms in a chain were repetitions, and the average density didn't vary all that much. So we were able to show that with using our PPIWordNet, we could get a lot more chains, but we weren't getting chains that were very rich, you know, because we really don't have, well, two things. We don't have enough concepts as yet, and we're only using some basic semantic relationships, basically is-a and part of. So we really need more rich semantic similarity measures. So that was the key findings, that we got significant improvements in the quantity of lexical chains, some improvements in chain quality. The size of the ontology increased by a small amount, but the resulting increase in number of chains went up quite a bit. So as I said, when you consider that overall we were adding in less than 100 terms to an ontology of 60,000 concepts, it really made the lexical chaining algorithm work much better. But the richness of the chains, in contrast to the quantity, was not very much by comparison. So it leaves open a lot of work for us to do. The first thing I'd like to work on, and this is a measure of how recent the work in this field is in biomedical information extraction, that it's only in the past week that the results have come available from the biocreative evaluation task, where people were testing out their information extraction algorithms on determining protein interactions. Those results have just been released. We now have the results available to us to say, well, how does our lexical chainer do at actually clustering lexical chains around existing protein interactions? Because we couldn't do that before. We'd like to see if, when we build these chains, are they spurious? Do they appear in places where they shouldn't, or do they actually cluster around interactions? Because that would be a very nice result to find, that we can use lexical chaining to detect protein interactions. And then secondly, to expand protein interaction WordNet. Right now we've got less than 100 concepts. What happens if we have 200, 1,000? It's a balance between being very parsimonious and having very well-defined and well-understood relations, or being very profligate and throwing everything into the kitchen sink. At what point do we stop getting more value? Better metrics for evaluating lexical chains, and the long-term goal, judging the validity of protein-protein interactions. How can we better help biologists find out whether protein interactions are real, but use tractable computer science methods to do so. And the acknowledgements are BioWebDoc project. You have to give a very catchy name to your project. Is Zhao Fenhei, who did a lot of this work with her master's thesis, Shadi Hassan, who was our information retrieval expert, and Gabe Musso and Zhou Yu, who were our PhD students in biochemistry. Thank you.
It worries me when you have a student or a person by the name of Shady in the thing.
No, it's Shadi.
I just wanted to say something. Years ago, you had a dream of being a speaker. Mm-hmm. And I've now heard you speak a couple of times at Waterloo, and you've achieved your dream.
Thank
made
you.
a very challenging topic very understandable, and I think that's remarkable. So
thank you. Well, you saw me back at the beginning. Too bad you should. Well, you did have that picture, that video.
I do have that. Someday I'm going to show it just to torture everybody.
Well, yeah, you should. It would be interesting to see the
She was an
before
egg
and after.
at the time, just a little tiny white egg, and we videoed it. But congratulations on that. Questions, comments? I'd like to get the microphone to you if you have comments or questions or thoughts about this yourself. I have one to start the thing going. As you know, my interest is primarily in the health informatics literature. And one of the real challenges we have right now is learning about evaluations at various levels of these systems. And the literature is spread all over the place. And there are many kinds of evaluations, from good to bad to terrible. And I wonder about the potential of applying these same ideas. I think it's a more constrained discourse analysis problem, but I'm not sure of that. But it would be incredibly valuable to be able to mine the literature for people doing evaluations of various kinds. I mean, I don't know, have you ever given thought to looking at the health informatics literature as opposed to biomedical literature?
Well, it's interesting. I started out in knowledge representation and then became sidelined in computational linguistics, and it was just very ironic that in order to do this computational linguistics project, I found myself going back and becoming a knowledge representation person again and getting steeped back in the work in ontologies. So yes, I'm quite interested in working on ontologies and health informatics, because it just seems there's just so much work that needs to be done, and the ontologies that are out there are just not very systematic, not very formal, not very easy to add to, and we do need these I came away feeling we do need these domain-specific ontologies, and there's such an enormous startup, to try and create them.
Just to pursue that a little bit more with the ontologies, obviously you basically did it manually.
Well, extracting the discriminating terms was done automatically. Finding the terms that were specific to protein interaction articles, we did that through information retrieval methods. But then constructing, like taking those terms and shaping them into an ontology, was done by domain experts.
Did you, or were you able to develop a vision of any support that you might create that would help you do it? I mean, it's a terribly labor-intensive task. And,
yes.
And the thing is, are there ideas of tools or methods that might?
I think methods, and I think tools. I think visualization tools would have been very helpful. building in a systematic verification too. I mean, we had two people who worked very well, and we built in the process by which they cross-validated and all through it. I mean, I think we did come up with some methods which could be used, but I could think of tools that we would like to have. You know.
Ming Lee published an article that we discussed regarding the same kind of thing. How is the work that they're doing different from what you're doing? Are there other approaches here, or are they just two examples of the same thing?
Which particular?
He did an article on protein-protein interactions. He was the final author. I don't recall the primary authors of that, but it was on protein-protein interactions detection. I know he's done a lot of work in algorithms, particularly related to.
Oh, well, our work is just is using is simply doing textual analysis. You know, I mean, when you think of biomedical information extraction, there's two branches of that. One is the actual genomics, and one is looking at the text. So we're looking at the biobibliome. You know, we're looking at the volume of text. So we're looking at what people write about who do the research.
I think he was looking at that too, but I'm not sure.
My field is pretty different. I don't know.
Yeah, put it close to you.
My field is pretty different, so this is just a little bit off the top of my head, but I know that when Google does Google search stuff, that one of the things that makes them so good at what they do is that they somehow have it frequency.
Yeah, that's what we did. We used that kind of ranking. But what we found was that we got a lot of spurious hits. You know, we came up initially with about 13,000 terms, but a lot of them were things we didn't want. In fact, we almost had to go through and rank them by hand because we got authors' names turned up an awful lot, or acronyms and abbreviations. So we had to throw away a lot of it.
A step beyond what I'm thinking.
Yeah.
Thank you.
So we needed to clean up a lot of that. But we did use those standard ranking methods. Yeah.
I know that there are different discourse theory available. When you're saying that you are using discourse analysis, do you use one of them, or does it make any difference to use one for biomedical, or pretty much do we need one for them, or we don't need create any more? Does it make any difference?
Well, lexical chaining is just a method in discourse analysis. It's not a discourse analysis theory. It's just a technique.
But does it also helpful to use like discourse analysis theories? There are so many of them available, and like maybe one of them it's like geared toward biomedical terms instead of using like causing and other stuff.
Well, I think that using computational discourse analysis in biomedical information extraction is a good thing. This is just one type of computational discourse analysis. You could use other forms. So lexical chaining is just one particular computational discourse analysis technique, and you could use others, and you would get different kinds of information. So this is just one particular kind of technique.
I think the question, though, is there some aspect, I don't mean to, I have a microphone, I don't mean to, if I'm saying the wrong thing, please ignore it, that there is a certain approach to the biomedical literature. I mean, people write differently than one would write love letters or something.
Oh, yes.
And is there anything about the biomedical literature, particularly where there's a type of discourse where a discourse theory that was close to that type of discourse would be appropriate? Am I getting at the same area?
Yes, I think so. But in fact, some of the work we're doing in another project I have, which is looking at citation links to understand the structure of scholarly scientific articles, and we're using rhetoric of science and rhetoric of biology. But that is very linguistic. It still needs a lot of work done to make it computational. But that is something that we could look to those discourse theories from the rhetoric of science.
I didn't hit the mark apparently. So you want to try your question again, or is that?
No, I was thinking most of the analysis I saw, they are looking like discourse, they are looking lexical chaining. It's not just for biomedical, even in your course that I was taking, there was like doing like summary of the newspapers, and pretty much they're using it. Looks like it's so widely used. Is it just because they're more promising?
Well, yeah, lexical chaining is nice because there are computationally tractable algorithms. The drawback is, as we found out, that you need a specific ontology to work with it. But the discourse analysis methods that have been, the computational discourse analysis methods have not been geared so far to the biomedical area. So there's an enormous area, of research waiting to be done. We looked at this past fall. I had a discourse analysis course, and some of the methods that are being used are applying legalistic structure, for example, to health informatics information, which it just doesn't jibe. It just doesn't work very well. So the people who are working in computational discourse analysis are still using a very formal structure and not geared specifically to the biomedical genre. So there's lots of work available if you want to jump into that field. But then again, there is a lot of work done by rhetoricians of science, but it's almost purely linguistic.
The other thing I'd like to ask you about is you pulled together an incredible multidisciplinary team. What were the challenges and things you had to do to make that work?
Well, actually, it was a joy. I'd say it was one of the best teams we worked with. I started out by doing a Google search, finding out who at U of T worked on information retrieval, information extraction. By then, I was led to Alan Davidson, who is a professor in biochemistry and works on protein interactions. And Alan put us in touch with students at U of T. Shadi had taken courses with me, and that's how I knew him, and Zhao Fan was my student. But I think the people and the personalities just meshed extremely well. And, we each learned something of what each other did. So, and we're still continuing to work together. And I have to say we thank the Waterloo Institute for Health Informatics Research for a seed grant, which funded this work. Which is true. We couldn't have done it without that, because this is how we afforded the research assistants to work on this. And next year, hope to put in a major grant proposal to continue this, because we now have a pilot system that came out of this. So.
Just one minor question is you had somebody in the name of St. Ange on there.
Oh, that was David St. Ange worked with Graham Hurst. That was on earlier work on how you score lexical chains, and we tried to adapt that, but it was. Again, as the comments made, like what's been done on lexical chaining, on discourse analysis, is geared to standard English. And their way of evaluating the strength of a lexical chain was really not appropriate for biomedical lexical chains. So I think we need to move computational discourse analysis into scientific writing.
Well, there are two, and I think that's an interesting thing, that's have a discourse analysis theory that is close to the biomedical type of writing. I mean, and type of. Now, the reason asking about St. Ange is we know Hubert St. Ange, who used to run Clerica, and I'm wondering if there's a relationship.
I don't know. I knew David, but I haven't seen him for quite a number of years.
Interesting to know that. And the other question was, he made me think of French. And has this been, have you looked at the potential of looking at non-English literature, which has some stylistic differences and so on?
Well, there is a EuroWordNet, for other languages, but I haven't myself done lexical chaining in other languages. I'm sure it's been tried. My interest has been in the scientific writing. I think my feeling is it would work pretty much the same, because the WordNet is meant to be a concept hierarchy, it's meant to be language independent, so it should work for languages other than English.
It's interesting because, sorry, Don, let me get you the microphone.
Just to follow up with that a little bit, English itself is very rich because it comes from so many different languages, whereas some of the other, we'll take French, for example, which is supposed to be highly structured. How highly structured it is, I don't really know. But one would guess that lexical chaining might be somewhat easier for that reason.
Well, computational linguists are very careful to make a distinction between concepts and words. And although WordNet looks like it has words in it, they're meant to be concepts that are represented by different words. So it's meant to be a language-independent linguistic ontology. You know? But it's confusing because they're words, but they're actually concepts. You know?
I had the opportunity to work with Chrysanne on the very early parts of what became the Inkpot, which she has now a full patent on, by the way, which is very, very remarkable, and to be congratulated for that. And I remember at the time it came out of the work, roots of the work in Germany of translation of technical texts from one language to another. And the other thing I really learned from that was that in different languages, you don't only use different words, you use different ways of explaining things. The comparison between German and English wasn't just the words or the idioms, it was how you explain things was different. Have you taken into account in the biomedical literature that maybe there are different approaches to explaining things?
No. I mean, there's a very rich body of literature in the rhetoric of science. It's fascinating, actually, about how people make knowledge claims in biology and science. I mean, much more so, as I said, in computational linguistics, we don't really write like scientists. Maybe not in computer science either. But, you know, in the biological and scientific fields, the articles are structured to make knowledge claims. They relate very closely to the style of the experiment. So, and the people who work in the rhetoric of science can tell us a whole lot about that. And that, I think that is something that we're following up on in our projects, on using that so using argumentation structure, using discourse analysis that is geared to the rhetoric of science.
Might be really interesting in our institute. We've talked about how we incorporate some of the work that Kathy Schreier's doing in rhetoric and work you're doing of having a group that looked at biomedical literature from those different vantage points, both find things in the literature and also to even detect problems in the literature, claims that are in fact incorrect.
That would be very interesting, and someone you should definitely include is Randy Harris, who is a rhetorician of science, and he's been our consultant in these matters. He's actually, well, you know Randy, he's part of Health Doc as well, but yes, I mean the way people make, what we've been looking at actually in the rhetoric of science is hedging. You know, that when people are putting forth knowledge claims and they can't actually say something very strongly, they hedge a lot. So by analyzing the hedging, you get a sense of how good the results are. And it's fascinating. But also, people hedge because they don't want to offend established researchers. You have a young person starting out who wants to get tenure, who doesn't want to knock some established person off their pedestal. They have to put their own claims out, but in a way that's very obsequious. And it's quite hilarious, you know.
Careful to destroy everything I've ever written. Chrysanne, thank you very much for the presentation.